Okay, so this is your book, The Basic Science of Oncology, 5th edition from Mackerel Education book I'm going to read. So the topic today is your cell proliferation and death. The cell proliferation has nothing to do because it's also, it contains only those how the cell divides, their regulation. So today's topic is about your apoptosis, the death of a cell. More precisely, the programmed cell death. So these are nothing but the cell cycle. We are here to discuss the death of the cell, a programmed cell death. So here we go. Modification of control of cell proliferation in cancer. The retinoblastoma is the first discovered tumor suppressor gene. And so cell death. The balance between the cell viability and cell death is critical for all the organism. If this balance get destroyed, then it will be fatal, and that leads to the cancer. So that thing has to be maintained, because every single time thousands of cells are being dead, more than thousands, sorry for that. So the thing is your some written highly regulated mechanism involved. Okay, so let's begin. The morphological changes associated with different type of cell death. Different type of cell death leads to two different kind of cell death. One is your programmed cell death, and the one is your non-programmed cell death, what we call necrosis. And the programmed cell death is your apoptosis. So here's two picture. Let me show you in this way. See better comparison between two cells. You can see the upper one is your apoptotic cells, the lower one is your necrotic cell, and the basic difference is written here in two color. So first, the apoptotic cell. The changes associated with apoptotic cell include rounding of a cell plasma membrane. Yes, it round. Necrotic cells associated with cellular changes are manifested by swelling of a cell. Yes, the cell is swelled. The organelles are also get swelled. Yes, I can. You can see swelled organelles also. But in case of your apoptosis, the plasma membrane get blebbed, and the plasma membrane blebbing occurs. Cytoplasm shrinks, and alternation of membrane asymmetry also occurs. Condensation and fragmentation of nucleus. So the nucleus is here fragmented. You can see the deep color is the nucleus, but here it remains cluster and all the goes lysis destroy. Okay, the cell at this stage of apoptosis become fragmented into apoptotic bodies. So cells become fragmented into apoptotic bodies, and the most important thing is the body is being eliminated by phagocytic cells without triggering inflammation. That's to be important. No inflammation occurs. Cell becomes bleb, condensation and fragmentation of nucleus. Okay, but in necrosis, the moderate chromatin condensation is also displayed in necrotic cells. More advanced necrosis are associated with disintegration of cell components and inflammation. The inflammation is the most important point. In necrosis, there will be inflammation. Okay. So the mechanism of apoptosis. First heading is your mechanism of apoptosis. So the first is discovered in your Sinorhabditis elegans. Loss of function, mutation of C ligands like your EZL1, SED3, SED4, gain of function, mutation, SED9 results in survival of this 131 program to die. So simple thing is that these pictures he want to tell. In C ligands, forget about this mammalian. In C ligands, the EZL1, which is an anti-apoptotic protein, it's keep on inhibiting this SED9 protein, and this SED9 protein function is to inhibit this SED4 protein. So, and the SED4 cause activation of the SED3, and that SED3 on activation leads to apoptosis. So here is shown that the loss of function of or mutation of C ligands in EGL1, SED3 or SED4 or gain in function of mutation of SED9. So SED9 if gain the function, it will lead to the program the cell lead to the death. That means loss of function of EGL1. So let's see if the EGL1 loses its function, it's unable to inhibit this SED9 protein, and as this SED9 is unable to inhibit it, it will express. As it express, what it will do? It will suppress those SED4, and but as it's unable to suppress in presence of your EZL1, but in absence it will be able to suppress. But in presence it won't suppress. So SED4 will cause activation of SED3 and the lead to apoptosis. So similarly, the SED3. If the SED3 get activated, then also apoptosis. Then your SED4 is also apoptosis. Here SED4 is activated, it leads to the apoptosis. Or gain in function or mutation of SED9. Here the SED9, if it gain its function, then it will suppress the SED4, so no apoptosis will occur. But if the SED4 gain its function, and this function to activate the SED4, so if it activates the SED4, it will cause apoptosis. So this all lead to the programmed cell death. Once again, EZL1, SED9, SED4, SED3. Okay. Now the apoptosis is triggered in mammalian cells in response to endogenous stimuli or exogenous stimuli. Apoptosis is also induced in response to inadequate cell matrix interaction, and the specific type of apoptosis is known as ankylosis. Forget about it. So this apoptosis occurs in two pathways. One is your extrinsic, that is your death receptor pathway. Another one is your intrinsic, that is your mitochondrial pathway. Anti-apoptotic protein like BCL2, BCLXL, they are the family of anti-apoptotic types protein found in mammals, and which are homologous to that of your SED9 protein. So in this picture you can see the EZL1 inhibits SED9, then SED9 inhibits SED4, SED4 activates SED3, that leads to apoptosis. So this EGL1 similar to bad big, SED9 BCL2, SED4 APOP1, and this is a caspase. Now come to the BCL2 family member and their role in apoptosis. BCL2 gene are homolog of the C ligands SAD9 as a prototypical membrane, so anti-apoptotic and pro-apoptotic. The anti-apoptotic protein is your BCL2, BCLXL. They are the anti-apoptotic. That means whenever they're present, they prevent the cells to undergo apoptosis. So here is BCL2. They're anti-apoptotic. That means if they present, they don't allow. They keep suppressing those APAP1 and that in Serigas you said 4. So by suppressing those APAP1, they will able to inhibit this anti-apoptotic apoptosis. So BCL2 family is your anti-apoptotic. In case of your pro-apoptotic protein, like I have discussed, this APAP1 is your pro-apoptotic protein that causes apoptosis. So those pro-apoptotic protein need to be get suppressed by this anti-apoptotic protein. These proteins share several conserved domains. So here is some picture I was not writing. The BCL2 homology, it has a BH1, BH2, BH3, BH4 domain. The BCL2 family members important role in regulation of the apoptosis. Overexpression of BCL2 in mammalian cells inhibits the release of cytochrome C as well as other factors important for apoptosis, like your AFF, SMAS, OMA, etc. from the mitochondria to the cytosol, obviously membrane. So overexpression of BCL2 also prolong to survival of these cells, increase their resistance to variety of this apoptotic stimulus, etc. etc. So role of caspase. The caspase, where it is, here your caspase, which leads to apoptosis. So the caspase is your cysteine dependent aspartate specific proteins. Cysteine dependent aspartate specific proteins. That means aspartic acid will be detected cysteine specific. The orthologous of the silicon protein is your SAD3. The first the caspases are present in a cytosol in their inactive forms, the procaspase or zymogen, to activate the require the proteolytic cleavage at the specific aspartate residues. So to activate the required proteolytic cleavage at that specific aspartate residues. These active caspases are heterotetrameric complex composed of two large subunit to small subunit. The inactive one is heterotetrameric. The active caspases are heterotetrameric. Sorry. So this heterotetrameric. Okay. So caspases are inactive forms. They have to be activated. They require proteolytic cleavage to specific sites, etc. Okay. So caspase unactivated, having this heterotetrameric complex to two large subunits and two small subunits. So caspase 8, 9, 10 are initiator caspase, whereas your caspase 3, 6, 7 are your executor caspase. The initiator one is your large domains and your executor has a small predominance and prodominance. Domains are different. And here is written subunits. They are a different thing. Okay. Caspase substrate including cytoskeleton, DNA damage repair, cytokines. So there are some caspase substrate, hundreds of types underlined. You can read out. Here's a picture. Your death domain from this fast file activates casp. Procasp, caspase 37, substrate apoptosis, whereas your signal like your intrinsic mitochondrial pathway, it includes the p53 gene activated that leads to the box cytochrome C release, formation of APF1 complex apoptosome, caspase 9 activated that leads to procaspase, initiator to executor caspase, and then apoptosis. You can read out all those things from this writing. Okay, here's a picture. Writing. Okay, so cytochrome C, Apoptosis, apoptosis. Three functional domain is there: N-terminal card, a central set for domain, long C-terminal WD450 for the apoptosis domains, etc., etc., etc. BCL2 homologous, BH3 only, anti-apoptotic, pro-apoptotic, BH4, BH31, BH2, BH etc., etc., etc. So here is your pro-apoptotic and anti-apoptotic. The pro-apoptotic is nothing, it is your, where's gone, man? Okay, the caspase exists in an inactive form. What do you call pro-caspase? So here is your pro-apoptotic, anti-apoptotic. Here is your pro-caspase. So BCL2 family member include both anti and pro-apoptotic protein. So anti-apoptotic is your upper one. Here like BCL2 anti-apoptotic, whereas your pro-apoptotic your FF1, caspase, etc. So the BCL2 family has anti-apoptotic like your BCLX, BCLW, etc. It has a BH4 domain, BH3, BH1, BH2, and a TM domain. And in your bags like your pro-apoptotic, it has a BH3, BH1, BH2, and a TM domain. But in case of BEAM, it has only the BH3 domain, like your BAT, Noxa, Puma, etc. It is the BH3 only. So it's because it plays only this single domain. And all those things of this caspase has some 4 subunit tetrameric, homotetrameric, heterotetrameric. So P20 is the large subunit, 2, and P10 is the small subunit. Caspase 1, 2, 4, 5, 9, 11, 12 possess card. Caspase 8 and 10 possess this death domain. And caspase 3, 7 nothing, just only this P20 and P10 segments. Then your death receptor pathways and your mitochondrial pathway in next video. Thank you very much.